A very warm welcome to all the participants who have joined us for this fourth inaugural session of BISMiS Live, the fourth year that we begin today. We have another set of 10 excellent speakers lined up for this year, and it'll be a pleasure hosting our first speaker for today. I am joined with Professor Faunus Venter and Professor Aaron Teo, who will also be co-coordinating BISMiS Live with me henceforth. A great thank you to them for volunteering to take up this responsibility. So you will be seeing less often of me and not get bored. We are also joined with the members of the executive board of the BISMiS Society. We have the honor of hosting our first speaker for 2024, Professor Svetlana Dedysh, who works at the Vinogradsky Institute of Microbiology at the Research Center of Biotechnology of Russian Academy of Sciences. The session will be a very interesting session on creating a bridge between microbial taxonomists and ecologists, using some options to increase the value of a microorganism description. This is an excellent topic that I'm very, fond of talking about, because as a taxonomist, many of the times we end up just describing the organism, but don't look at the application part. And Professor Svetlana is an expert on that. So Svetlana received her PhD in 1990 from the Moscow State University and joined the Laboratory of Soil Microbiology and Biokinetics at the Vinogradsky Institute of Microbiology, where she started her research on methane oxidizing bacteria in northern wetlands. The research was carried out in a close collaboration with the Center for Microbial Ecology at the Michigan State University, United States, and the Max Planck Institute for Terrestrial Microbiology, Germany. In 2005, Svetlana defended her doctoral dissertation on acidophilic methanotrophic bacteria, and since 2008, she is the head of the Lab of Molecular Ecology and Phylogenomics of Bacteria at the Vinogradsky Institute. Her main field of research is the microbiology of northern wetlands and bacterial systematics. Areas of research expertise of Svetlana include biology, diversity, and ecology of methane oxidizing bacteria, characterization of bacteria from poorly studied phyla such as the acidobacteria and the planktomycetes, microbial diversity in sphagnum-dominated wetlands, cultivation, genome analysis, and taxonomic description of novel bacteria. She is an author of more than 170 research and review articles. Since 2014, she is also a trustee of the Burgess Manual Trust and one of the editors of the Burgess Manual of Systematics of Archaea and Bacteria. She was appointed as a member of the ICSP, the International Committee on Systematics of Prokaryotes, and served as a member of the Judicial Commission of the ICSP since 2014 until last year. She is also an ambassador of the International Society for Microbial Ecology in the Russian Federation. With this brief introduction, I'll request Svetlana to begin her presentation and share her screen.
Okay, good morning, everyone. And today I would like to share with you some of my experience regarding how to make taxonomic descriptions more interesting for people who stay outside of the taxonomy field. In my opinion, this is very important thing because taxonomists are sort of special people. On this slide, I used two cartoons to illustrate sort of a difference in between microbial taxonomists and microbial ecologists. And I should give a credit for this cartoon to Marta Trujillo, who already used this cartoon in her presentation on BISMiS Live. So who these people are? Actually, they're supposed to describe existing microbial diversity, maintaining precise system of nomenclature, and to study phylogenetic relationships of microorganisms. Some people, some scientists view microbial taxonomists of being very formulaic people who are busy with giving new names to new microorganisms without really going deep into their biology, ecology, and other features. By contrast, microbial ecologists, they deal with natural ecosystems, and their function is to explore diversity, distribution, and functional role of microbes in the environment. But of course, they also deal with microbes, and they use all pool of knowledge which is produced by microbial taxonomists. I cannot imagine any microbial ecology who would describe diversity in the ecosystem without using names, genus, and species level names, and also other names, which are basically were proposed by microbial taxonomists. So I assume, and everyone can assume, that there should be a linkage, close linkage between these two groups of scientists. But in fact, it's not always like this. When I was preparing to this presentation, I screened a number of publications from journals which are listed here. These are journals which microbial ecologists use to publish their papers. I wouldn't say these are all existing journals, no, but I wanted to do some quick search. And these are nice journals, you can see it. And I looked for the proportion of articles which cite descriptions, taxonomic descriptions of individual organisms, which were discovered in these ecological studies. Yeah, for example, all of the studies, you know, these are typical pictures that are published in ecological surveys, like, you know, composition of microbial communities in some particular habitat, analysis of shared and unique OTUs, and also some network diagrams showing how microbes interact with each other. And of course, they all these papers are full with names of various microbes. And the question is, if these people who produce these papers cite taxonomic descriptions of those people who, you know, isolated, described these microbes and gave them names. So the answer is here in this simple diagram. So I was surprised to see that 70% of people of this environmental microbial ecologists do not cite any taxonomic papers. So yeah, 30% do that. 10% in 10% of these papers, only one taxonomy papers is cited for 100 of other citations. Other papers cite a bit more, up to 20% of all references in the list of references are taxonomic papers. But I have to say that the authors of these papers commonly have experience in taxonomic descriptions as well. So if you want to see it in general, then most microbial ecologists either do not cite taxonomic papers or cite very few of those. So what is the reason? Is this a reason with microbial ecologists, or maybe this is a reason, the problem with us? So let's think from the point of microbial ecologists. What kind of information they expect to see in taxonomic papers in order to be cited in their study? So first of all, the reason for this incompatibility between taxonomists and ecologists is a low informative value of abstracts of taxonomic papers. Of course, we all know that we are not given much space to write an abstract about some particular organism. And yet, the abstracts which are routinely published report very few features which are of interest to microbial ecologists. I included some of these features here. These are basic features of commonly reported set of traits. Isolation source, of course, is of interest to microbial ecologists, but gram-stain reaction, basic cell shape, and also various indices like 16 sRNA, gene sequence similarity, DNA, G+C content, various nucleotide and amino acid sequence identities, fatty acid composition, and also type strain numbers in culture collections. Of course, everything what is in gray is not of interest for microbial ecologists. Yeah, okay, there is also a type of metabolism and relation to oxygen, which in most cases are reported in abstract, but not always. You know, you would be surprised, but if you screen some papers, you would not see this information in there. So more rarely reported features are relation to pH and temperature, growth substrates, and specific functional capabilities. But the latter are cited only if, for example, we are talking of nitrogen-fixing bacteria, methanotrophs, methanogens, phototrophic bacteria, and so on, right? So this is not always the case. So then if we go to the manuscript itself, then of course we will see the lack of information regarding ecological traits of newly described microorganisms in the manuscript. What kind of info microbial ecologists would like to see? So here in red, I listed some of these features or some of this information. And of course, every ecologist would like to start with habitat characteristics. So not just soil near to some city, but characteristics of the soil, at least very brief ones. What is the pH? What is the content of organic matter? Then, of course, some interesting information is description of approach which was used to isolate this microorganism. Of course, if this is not just a common plating on organic complex media. Cell motility, morphology, and also ultrastructure are among the features which may be of interest. Relation to oxygen, pH, temperature, growth substrate, growth rates, specific functional capabilities, and of course, specific environmental adaptations. We will walk through these features later on. These are only some sort of comparison what ecologists would like to see in taxonomic manuscripts and what they see in reality. So what they want to see is indicated by red. So this problem was recognized long ago. For example, Cowan wrote that taxonomy is written by taxonomists for taxonomists, right? So that was written in 1971. Ramon Rosella Mora wrote in one of his manuscripts that even taxonomists might get bored when reading many of the hundreds new descriptions of bacteria and archaea published each year. So, this is written by someone who also described many interesting bacteria, and so the person who recognizes this problem very well. And I would like to attract your attention to one of the papers. Actually, several papers on this subject were published lately, but I would really recommend you to read the one listed here. It was prepared by 3 chief editors of leading taxonomic journals, Ian Sutcliffe, Ramon Rosella Mora, and Marta Trujillo. So here's the information. It's relatively fresh article published in 2021. And this excellent paper gives all recommendations to microbial taxonomists about ways to prepare taxonomic description that would sort of up-to-date it to needs and requests from the side of microbiological community and also from ecologists as well. What about myself? Why do I care of this issue? Kamlesh already introduced me, and so maybe some of you already catched why I am so much interested in this linkage between microbial taxonomy and ecology. But the more information is here. I basically work in Vinogradsky Institute of Microbiology, and my supervisors were Professor Panikov and George Zavarzin. Both were very much interested in biospheric functioning of biospheric role of microorganisms, and always looked at microbial diversity from the standpoint of functions in the biosphere. Then I also have a nice experience of working in Center for Microbial Ecology in Michigan State University together with in the laboratory of Jim Tiji, and also in Max Planck Institute for Terrestrial Microbiology in the department of Professor Conrad and laboratory of Dr. Werner Lisak. You see, all of these people have deep expertise in microbial ecology and the authors of many hundreds of papers that link microbial identity and microbial function in the biosphere. This is my background, and this is why I really would like to see more papers, more taxonomic papers that preview some information which is of interest to microbial ecologists. So myself, I recently published a review article in Systematic and Applied Microbiology, and here's this information, basic information about this publication. As you see, this paper deals with difficult to culture bacteria, but the information provided in this article is relevant for describing all kinds of microorganisms, no matter if they are difficult to culture or not. So basically, some advices which are given in this article are useful for many people who stay in the field of taxonomy. So, okay, let's walk through these advices. Where to start in the taxonomic paper? My advice is to start with significance. So basically, first of all, you should explain why this organism, which is described in your manuscript, is of interest in principle, yeah? So what could be the cases? For example, it could be very abundant in a particular habitat, or by contrast, it could be widely distributed in various ecosystems, sort of cosmopolitan organism. There is a very interesting situation, of course, of interest to many people when the very newly described microbes belong to as yet uncultivated phylogenetic group. So it's even no need to give some advices in this particular case. So the paper should start with this early uncultivated group. So, but there could be many other cases. And as the example, I can give you an example of our paper, which we described new acidobacteria, which are very widely distributed in northern terrestrial habitats, in wetlands and also in soils. And this was case with tundra habitats, which are covered by lichens. And when we examined microbial diversity in this habitat, we found that acidobacteria are numerically abundant group or dominating group, and the isolate which we received in pure culture represents a significant part of acidobacterial community, right? So this is another case when we worked with wetlands and examined populations of planktomycetes, which are very abundant in wetlands. So on this map, you see the wetlands which were peat from these wetlands was used for the analysis. Both raised wetlands, raised peat bogs, and forested fence we used for the analysis. And here is the outline of microbial or bacterial diversity in these wetlands. Yeah, not the whole diversity, but just planktomycetes. You see these yellow bars. These bars indicate the group of planktomycetes which was not obtained in the pure cultures before our study. And we've got one of the strains that represent these numerically dominating groups of planktomycetes. So this is another example of where to start your taxonomic description of the isolate you have in hands. So, of course, other characteristics which are of interest for microbial ecologists are habitat specifics. Yeah, I already told that, for example, when you state that some sort of isolate was obtained from soil next to the city of this and this point, this approach is completely wrong, right? So when you say this isolate was obtained from soil, you have to explain what kind of soil is this and what characteristics of this soil, which may influence, for example, of course, microbial diversity in this soil. If possible, you should also state the temperature during sampling, pH of this soil. Basically what is written here has wider meaning, not about soil, but all ecosystems in principle. So the factors which should be described are temperature, pH, salinity, availability of oxygen, availability of organic carbon, and so on. So, you know, the really rare case is when people really pay attention to availability of oxygen. For example, people describe deep marine sediments and describe isolates which was obtained from marine sediments. And suddenly you see that authors state that this is aerobic bacterium. And then what should you think of this story? Basically there should be no oxygen in deep marine sediments, and yet the isolate is described as being obliquely aerobic bacterium. So cases of this should not be, basically published, because referees should really take care of looking into these issues. And nonetheless, so of course, it could be that you isolate aerobic bacterium from deep marine sediments, but then you should say from where this bacterium received oxygen in this habitat, or maybe it just was traveling from upper layers of the ocean to the deep layers, and it's not living there, but just, got there together, sort of sedimented with some organic particles or something like this. So basically, it's necessary to pay attention to these issues. Then if you use some interesting approach to obtain the microorganism in a pure culture, you should definitely report this in your manuscript. In some cases, this organism is difficult to handle. For example, it produces cell clamps, or maybe it forms biofilms. All these features should be reported. Then we come to cell morphology and cell ultrastructure. In most papers, the morphology is given, in a very brief paragraph, like cocci, rods, yellow color, non-motile, and so on. That's it, what you can get from this description. And yet, morphology of the microorganism have very big significance for understanding what this microbe can do. So basically, it's good to focus on specific morphological features, pili, fimbria, flagella, capsules, S layers, and maybe outer cell vesicles. The figure given here was taken from a very nice review of Young. Actually, he has two very nice reviews published in 2016 and 2006, which shows a really a nice linkage in between cell shape and functional consequences of this cell shape, which give sometimes competitive advantage or helps or ensures survival in the ecosystem where these microorganisms came from. So the shape itself gives you some tips for studying this microorganism, and I would really advise you to read these nice reviews. Just one example. This nice bacterium was isolated from peatlands. This is methanotroph, spiral-shaped methanotroph. And of course, we could describe it like, like spiral-shaped methanotroph. That's it, point. Yeah, but we thought that we could do more. And we examined the advantage given to this methanotroph by this spiral shape. So, and the finding was really fantastic. So it turned out that this spiral shape gave a specific advantage of reaching the substrate of this microbe, methane, in environment, in a heterogenic environment, yeah? So this bacterium just goes into peat or sediments easily because it moves like a cork bottle opener, like, yeah, cork opener, and reaches the target site where methane is available very fast. So here there is experiment which shows that cells from these spiral-shaped methanotrophs, they were added here into Petri plate in the medium, were able to penetrate very fast through semi-liquid medium. This is semi-liquid medium prepared with agar, and in some aspects, in some additional experiments, we also added some mineral particles in this semi-liquid medium. So, and we're moving towards methane availability, and methane was kept here. They did it extremely fast. So, and this competitive advantage was given by cell shape. Yeah, so paying attention to cell shape is very important. Okay, you say, but yeah, these are extraordinary cases. And myself, I have a simple bacterium, for example, spherical shaped. Yeah, okay, we also did. We also had, of course, some bacterial with spherical shape. This is one example. This is a planktomycete called Fimbri globus ruber. And yes, if you look at the microscope, phase contrast microscope, you see spherical cells, which somehow tended to form cell clamps, aggregates. So it grew only being highly aggregated. But when we examined this bacterium under an electron microscope, we found many fibrils or pili, and cells clamped to each other by means of this pili. And what was the explanation which we found later on? So these pili are used to adhere to some solid substrates, like, for example, biopolymers, such as chitin. And this microbe uses chitin as a source of carbon and energy. And of course, when chitin is not available, cells simply clamp to each other, yeah, and grew like this in a medium. But there was a big sense in looking into this pili and their function in the environment, right? Okay, then importance of cell ultrastructure should also not be underestimated. There are very few papers currently, taxonomic papers, which report the results of studies of cell ultrastructure. And believe me, people who do that, they lose a lot of information. So here's one example of our studies. We worked on acidobacterium, which I already showed you before, was isolated from lichen-dominated tundra soils. It had, sort of interesting cell morphology. Many cells clumping together, forming aggregates. But when we looked at the ultra-thin sections of these cell aggregates, we found very unusual picture. So the cell is here, and what is around is a specific cell sacculi, which are made of tiny, tiny fibrils. You can see it here. So more or less, cells live without sort of houses built on some sort of material, which is produced by cell themselves, by these outer vesicles. So these fibrils originate here and then transported by vesicles to the outer space of this bacteria. And when we examined these bacteria with X-ray analysis, we found that these houses are indeed composed of tightly packed fibrils. These fibrils were packed parallel to each other. And when we decided to address this issue by looking at the genome sequence of this bacterium, we found that it's capable of cellulose biosynthesis. So of course we examined this feature using genome analysis and confirmed this finding by applying cellulase to these aggregates. So these are original cell aggregates, and when cellulose was added, they fall up on individual cells. So this is the case when examination of ultra-thin cell sections gave us a further navigation, and we revealed very interesting features: cellulose biosynthesis to create the specific cell houses which protect the cells from harsh environment, and also the ability to produce cellulose. So this is another example which I would like to share with you. So this is a thin section of the planktomycete. I talked about this planktomycete before. It was isolated from wetlands. And also, these are simply coccoid cells. Nothing very special. But ultra-thin sections showed us the presence of sort of bacterial microcompartments here, and some are visible here. And when we played with different substrates, we found that these bacterial microcompartments became dark when the microorganism received not the glucose, but some more environmentally relevant substrate like rhamnose, which is a part of the some polysaccharides that are the parts of bacteria of plant cell wall polysaccharides. So, and these bacterial microcompartments, of course, were known before, and they are used in these cases when the substrate being oxidized turns into some compounds which is sort of toxic for the cell, for example, aldehyde. And this is the case when these plant-derived polysaccharides are being oxidized by the cell. Again, we had a look at the genome sequence of this planktomycete and found a route for rhamnose metabolism in this bacterium. And one of the toxic products here is lactaldehyde. And this is why we saw these bacterial microcompartments in these cells. So once again, we would never look for these details in the genome sequence, but examination of ultra-thin cell structure gave us this hint to look closer at the substrates utilized by this bacterium and the machinery which is used to utilize these substrates. So a very important characteristic, which is always important for microbial ecologists, is growth substrates and growth characteristics. So I am not a fan of biolog plates. Of course, this is fast and relatively cheap approach, but the information is close to zero. Yeah, okay, you can examine a wide range of sugars and some other compounds, but these substrates are of low interest for those who deal with ecology of microorganisms. So I believe that each taxonomic study should include testing substrates which are indeed relevant for native habitats of the organism you've got in culture. Yeah, they could be various substrates, for example, biopolymers or what is available in the habitat, I don't know, hydrogen, methane, and some other substrates as well. Yeah, then the ability to utilize these substrates should be also measured somehow. You know, it's not just plus and minus in a table. But if you find some optimal substrate which is preferred by this microbiome, you should try to examine specific growth rate or doubling time on this substrate under optimal conditions. Yeah, one of these examples is shown here. For example, the acidobacterium, which I already talked about, turn out to be capable of growth on chitin, one of the biopolymers. And what's the linkage to natural habitat? The linkage is that this microbe was really abundant in lichen-dominated tundra. So, and of course, the dead biomass of lichens is a rich source of chitin that could be degraded by other bacteria. And this acidobacterium was capable of doing this. So what you see here are images of chitin microparticles densely covered by this acidobacterium, Sarcina. And this is fish images of this acidobacteria. So this is a linkage explaining why this bacterium is so much abundant in this particular habitat. Again, then we come to chemotaxonomy and the value of lipid analysis. So this is just a picture. I even don't want to say what strain was this. There are two strains, actually, one and second. And we analyzed lipids using some conventional analysis, and we received results which many people receive and even include in taxonomic studies. Yeah. So what was the result? Number of aminolipids and phospholipids, and only one of them was identified in this study. Yeah. All others were addressed as unidentified aminolipids or unidentified phospholipids. Yeah. So the informative value of this figure or this part of your work is absolutely close to zero. So basically, you said nothing. So in my opinion, this information should not be included. What to do? Well, the best way is to identify these unique lipids, but of course, not everyone could do that. We were lucky. We had very nice collaboration with our Dutch partner, Professor Jaap Sininge-Damste, who he and his laboratory are top experts in lipids of microorganisms. And for example, they identified unique methyl-ornithine membrane lipids in planktomycetes, which we isolated from northern wetlands. This was very interesting discovery, and so one of the explanations why these bacteria have these lipids is that phosphorus is simply not really available in oligotrophic wetlands, and this is why methyl-ornithine lipids are generated by this bacteria or produced by this bacteria. So, probably they're also responsible for maintaining rigidity of cell wall of planktomycetes, because it's very difficult to lyse cells of this bacteria. This was some extra work, of course, but since we identified the presence of these lipids in planktomycetes, we included this information in each of our taxonomic descriptions of a novel plankton mycet. And what's the benefit for microbial ecologists? Yeah, for example, you can trace specific distribution of this particular group of microorganisms in the environment by means of extracting lipids from environmental samples and quantifying these lipids. This is an example of the paper which was produced with the use of this information. And finally, we come to the point of linking information about microorganisms' identity and its particular habitat. So how this information can be incorporated in ecological studies. So here's one example of our investigation, which was done in Tundra. So basically the interesting thing about this habitat is that this is basically a gradient from absolutely unfixed sand, not covered by any mosses or plants. Then it is changed by semi-fixed surfaces, semi-fixed mostly by lichens, with some mosses as well. And then with some well-developed plant vegetation, higher plants and mosses as well. So there was a nice gradient, and microbial community was examined, the shift in microbial community between these three eco-niches. So this is a standard diagram, which is produced by our partners in this study. They are soil microbiologists, of course. They made a profiling of these communities by 16S gene fragments and found diversity, and that acidobacteria are one of the most numerically abundant components of the microbial community in this habitat. And this and many other studies stop right here. So people simply don't go deeper in the analysis of this data, which are in fact available. So we try to go deeper. So we extracted the pool of acidobacterial reads from the pool of data received and tried to analyze them. So this is a gradient. See, unfixed sand, then semi-fixed sand, and this is mature soil. You see there is a gradient of acidobacterial diversity, a clear gradient, yeah? Acidobacteria that dominate in unfixed sand then disappear when soil is already in a soil which is rich in organic matter, and other bacteria which were not present in the initial stage of this vegetation pattern, of development vegetation pattern in tundra, they appeared later on. So basically we analyzed this diversity of acidobacteria according to some groups, and you see the gradient. And then we went to operational taxonomic units and plotted them in this network, using this network analysis. And you see again a clear gradient, some operational taxonomic units, which are numerically abundant here in a sand, unfixed sand, disappear here, but then other operational taxonomic units appear. So, and where's the information, you could ask me? Yeah, the information is here. You simply have to retrieve it. And the knowledge of taxonomic descriptions will help you to do that. For example, look at this operational taxonomic unit. Red arrow point to this taxonomic unit. And this OTU is shared by lichen-covered surface and mature soil. What is this? This is acidobacterium, which I told you about, which is typical for lichen-covered tundra, which can degrade chitin, the major component of lichen-derived biomass, right? So makes a big sense, yeah, that this operational taxonomic unit appears here and stays here as well. And what are those operational taxonomic units which are pointed by blue arrows? This is not Acidobacterium of the genus Granulicella. These were isolated from mosses, and they're also capable of degrading some components of biomass of high plants. So they're typical for mosses and for plant colonized surfaces. See, if you address some taxa which appear in your analysis and look into corresponding taxonomic paper, you can link basically the molecular ecology study and taxonomic study. And I would recommend you guys that you at least try doing this with some of your studies or samples, because you may find very interesting patterns. So basically, we are close to conclusions. I believe that the practice of preparing taxonomic descriptions that report only a minimal set of features to name a newly isolated organisms makes this gap between microbial ecologists and taxonomists bigger and bigger. Yeah. So, and basically, we should stop this. I would say that you should not hesitate to invest time in detailed analysis of cell biology and experimental verification of genome-encoded capabilities of newly isolated microorganisms. You may say, well, it takes a lot of time and also it's not easy. Yeah, I agree with you. Going this way is difficult, yeah, because preparing such a interesting taxonomic description may require several years of work. But nonetheless, those who would be able to go this way will be rewarded because there will be a knowledge in their manuscripts which is of interest to a broad microbiology resource community outside of taxonomy field. And this is something which is not about the microbe only. This is something about your scientific reputation as well. And I think this is very important. So, and on this note, I would like to thank you for your attention and to stop my presentation. Thank you.
Thank you, Svetlana. This was really exciting and very intriguing to see that how taxonomists describe and limit themselves to describing organisms based on minimalistic approach rather than a extensive approach of digging deep into the physiology of the organism.
Yeah, that's true.
Yeah. Thank you so much for this enlightening talk. We've received a few sessions and few congratulatory remarks as well. The first question that comes is from Dr. Jugsharan Singh Virdi. He's asking, don't you think that pathogenicity is also a sort of an ecological attribute of the microbe? And we need some preliminary data on this attribute also about any newly described organism. And he also congratulates you on your excellent talk.
Yeah. Thank you for this interesting question. In fact, I was going to say from the very beginning of my presentation that probably I would not navigate any people who work with medically related microbes. Yeah, because what I was showing was purely about ecology. But of course, you are right to some extent, because this information may be also of interest for microbial ecologists. For example, if we talk about microbes that are phytopathogenic, yeah, colonize some plants, for example, or feed on some insects or whatever, right? I would say then this feature becomes the most important one. And if I would produce some manuscript, I would really focus on this particular trait because this is the most important one, right? So my big advice to everyone is that when you prepare a manuscript like this, first you think about title, because title of this paper should already give a message to the rest of microbiological society, right? Why this microbe is interesting. And second, pay attention to the abstract. And of course, pathogenicity, if present, should be a major highlight of this study. Of course, yeah, I agree.
Yeah, good. I have two very specific questions that cropped up in my mind while listening to your talk. You might find that they are kind of, off the talk session, but they are kind of related for me. Um, we heard that the IJSEM editorial board is working on a new format for taxonomic descriptions. For the IGSEM and other, for the community in general. I wonder how much of this additional discovery type approach is included in those taxonomic description formats by the IJSEM. Is there any recommendation on including these characteristics? That's the first question. I'll take the second one after this.
Mm-hmm. Look, I'm not a member of the editorial board of IGSM, but I'm one of the editors of Systematic and Applied Microbiology Journal, right?
Right.
And this practice is already implemented, starting with this year. Maybe some authors already faced the situation when they sent the manuscript to Systematic and Applied Microbiology and then received a recommendation that they basically need to include more data on environmental significance of this organism, for example, to include some metagenomes obtained from the same sample from where you receive your isolate into your phylogenetic analysis, right?
Right.
Or produce some other data which would demonstrate that this organism is either numerically significant or possess some function or whatever, right? So I think that Ramon already announced this during last BISMiS meeting and illustrated this with some examples. So this is already on the way with systematic and applied microbiology. So with IGSM, unfortunately, as I said, I don't know, but I may imagine that things like this will be implemented as well, because many editors of IGSM are also interested to see a deeper linkage in between microbe, which is described, and the habitat from where this microbe originated. That would be relevant, you know. I think this is a very good idea. You know, because sometimes I talk to people who work in taxonomy field only, and they spend, you know, the whole life describing new microbes, and then they're even offended when they see that some microbial ecologists, you know, use the names of the microbes they described and did not cite the publication. Yeah. And how would you you know, react if you would spend several years of work to describe something in a very nice way, and someone takes the name and doesn't give you a credit for this? Yeah, this is something which shouldn't happen as well, right?
Yeah. Yeah. So luckily we have Aaron with us, Aaron Oren, who would like to add from the IGSEM perspective. Aaron, you are live.
Not only from the IGM perspective, from my own perspective, let's call it like this.
Okay.
Thanks, first of all, to Svetlana for this wonderful talk. And I must say, I agreed with every single word you said. I, during the talk, noted 4 comments. First of all, about your first slide about the abstracts and the information found in taxonomic abstracts. You are, of course, right that there is very little ecological information in those abstracts. But as you said rightfully, these abstracts are limited to 200 or 250 words. So they give mostly the taxonomic information. Then you made a very important comment is authors should explain why a new organism is interesting and important. And I see this all the time. As you know, I review every single name of new species, new genera, etc., that are proposed. And very often, prospective authors contact me before they write their paper, and I always like this. And I very often see something like this. Dear Dr. Oren, I have a new Pedibacillus or a new Pseudomonas or a new Halomonas or whatever. Can you please give me a name for it? Then I ask, what is so interesting and special about your new organism that special features that I can use in finding a good name? And quite often the answer is, I really don't know. There is nothing really special, but the ANI value of my organism is less than 95% with the closest relative. So I have to propose a new species. And then I continue this discussion and ask, why are you doing this? What is so interesting? And either the author doesn't reply or they say, okay, my supervisor told me to describe a new organism from this single colony that they have. So this is the kind of descriptions that we see all the time, unfortunately, but there's little that you can do about it. Comment number 3 about these minimalistic descriptions. Here, it's of course true. A week and a half ago, the publications committee of the ICSP, which governs also the IGSCM, of course, had a long discussion about this. And we know that the problem exists. If you read the rules of the code, the International Code on the nomenclature of prokaryotes, Rule 27 tells you that there must be some kind of a description, but it does not tell what this description should contain and what kind of information should be present. And that's rightfully so, for this is a code of nomenclature only. It means that every journal and every journal editor has the freedom to decide this is, yes, a good description or not a good description. This includes also editors of IGSCM. And there was a very clear recommendation to IGSCM editors, and this information will be forwarded to the editors very soon, that don't be afraid to reject papers that have very poor descriptions. So I expect that quite soon there will be new recommendations to the editors about the kind of descriptions we would like to see. And indeed, ecology is a very important part of these descriptions. And my 4th and last comment is about those long lists of substrates from biolog plates or other miniaturized systems. In a recent paper that I got on my desk, again, to check the nomenclature for IGSCM, I saw 2 descriptions that were each about 2 pages long, with 200 or 300 substrates from these miniaturized systems. It is, in my opinion, completely useless. These substrates, the organism in its habitat never encounters those substrates. Most authors who list those substrates don't even know what these substrates are. If you have a description with 200 substrates that are used for growth, and that's what authors very often say. No, biolog doesn't check for growth. It just says about something about the metabolism that is increased when you add a substrate. It's not growth. So I agree that this is absolutely useless and in addition, if you have one colony, one strain, one new species, having a list of 200 substrates, the moment you have another strain that is deficient in one substrate that the type strain does not have, it means that you have to publish an emendation of the description. On the other hand, long ago, I had a discussion about this with Peter Kempfer, who before Marta and before me was editor-in-chief of IGSCM. And Peter Kempfer very clearly told me, the more information about the type strain there is, the better it is. So he was very much in favor of listing all those uninformative substrates that biolog plates or other miniaturized systems gives you. So some people apparently like this. I don't.
Thank you, Aaron. Svetlana, you would like to add something?
Yeah, because I see two other questions appearing here.
Before I go to that, I'll take one question that I wanted to follow up. I clearly see a very high regard and the importance of the ecological features that need to be described along with the description of microbial species or new strains. I'm confused here in the sense that this is a little bit uplinking. When we propose that C code is a code to describe organisms from genomic sequences, I see a limitation there in the sense that if we describe organisms from only genome sequences, how much of information, which is the real life information of these organisms, will we be able to add to these taxonomic descriptions when we describe organisms from only genome sequences?
So I should answer this question.
Yeah, I mean, I would like you to add whether what I'm thinking is correct or is it wrong?
It's very difficult to analyze a substrate utilization pattern of a particular microorganisms based on substrate utilization, based on genome sequence,
Right.
It's nearly impossible, yeah?
Right.
For example, in my laboratory, we have really an expert guy who examines glycosyl hydrolases for the whole his life, right? And the situation with some easy substrates is obvious. You can identify them. But as soon as you go to some specific compounds, like even with chitin, the situation is not so clear because, there is something which can maybe be a chitinase, but there is no phylogenetically related enzymes which have experimentally confirmed activity, in each of the cases which I showed, we had to clone this enzyme and to confirm this activity so that the referees accept this as a statement, everyone knows that many genes are sort of coding something which is not known, right? So I don't think that genome analysis may help to make a spectrum of substrates utilized by the bacterium. May give some hints. For example, there is a cellulase, or maybe putative chitinase, but not more than this, Much easier when you work with genome of methanogenic bacterium or methanotrophic, or, for example, like phototrophic. You can make some strong conclusions because there is a lot of reference sequences which allow you to do this firm statement about what type of metabolism this bacterium has. But with some other bacteria, these are nearly impossible. So I believe that descriptions which will be, fixed via the SEC code will be by far less complete than those based on examination of pure culture, right?
Right.
So someone who will isolate this bacterium later on would have to fix up these gaps in the original taxonomic description, which was published in the SEC code. I don't know if people will be motivated to do that or not. Yeah, because the bacterium was already described, name is given, so a big impact factor is already in the pocket of someone, not yours, right? Why should you spend your energy to fix mistakes made by some other guys? Maybe some people will do that, maybe not. Let's see. Let's see.
Thank you. Thank you. I'll move to the questions coming from the participants. An anonymous attendee is asking, a strain isolated is usually different from the same strain domesticated in the lab. How do you deal with this situation? There are no more worries about it.
Yeah, of course, you should always keep that in mind. And of course, we're aware of this situation. Yeah, you isolate the bacterium. At the beginning, it doesn't want to grow. It produces a very, very poor growth. But then later on, it becomes sort of domesticated. Yeah, if you are able to figure out what are the optimal growth conditions and what are the best substrate for this bacterium. So I don't think we should worry about this. But of course, the specific growth rate, if it is reported in the taxonomic manuscript, may be different to that this bacterium has in native habitat, right? So we should simply keep this in mind. That's it. But I would not really worry about this fact.
Another side of the same question is, in your own experience, how many of these difficult to grow organisms, once they have been cultivated in the lab, have actually grown like any other organism in the lab?
Not like other organisms. Of course, they will differ. They will remain different, you know. But thing is that in the beginning, it may be very tiny colony, yeah, which require maybe several months to get grown to some amount of biomass you can play with, right? And then if you work with this organism in the laboratory for several years, yeah,
Right.
It may start growing in a reasonable way. But of course, it will never grow like Pseudomonas or Arthropteryx or Bacillus, never, ever. So it may grow like 3, 4, 5 times faster than in a stage on isolation. But it won't get close to fast-growing organism because it has completely different life strategy. It shouldn't get to the team of fast growers.
Right. Yeah. The next question is from Preben Nilsson. The question is, does it make sense to describe a species solely when the genome sequence is the main source of information disclosed? Do you see a value of having type material in the form of a physical viable strain available?
You know, does it make sense or not? It's just a question of, you know, creating a system for molecular ecologists who would like to give names to metagenomes, well-examined metagenomes they produced, right? And also to prevent a chaos of chaotic creation names by microbial ecologists. You know, some of them even don't know that there is a code and a strict rules, but they keep creating names. So, of course, maybe there is not much sense. But I think we will see. SEC code is already start functioning, and to my understanding, several manuscripts were already published in Systematic and Applied Microbiology, describing new species and new genera. And I also remember that Ramon told us on the last meeting about papers produced by his research team. Few species of a newly described genus were described based on isolates. I mean, in the code, with a code, a bacteriological code, but one which they failed to deposit in culture collection was described via the SEC code. Yeah. So we will see how does it work and if these systems could work together. So no good answer for you. Sorry.
Fanus has raised his hand. Fanus, you would like to add?
Yes, I would like to come into this conversation because this is something that I quite like. But over the years, I've seen that there are two different streams of thoughts, and I sort of support both of them. And people will know that I've always said that we need a way to describe species very quickly, just give a name and have a pin on the map. That's all we need. And at the moment, I think part of the problem is that we give too much credit to those people just putting a pin on the map. What we want, but it is important that we put the pin on the map. Otherwise, we lose information. So in my mind, there are two ways of describing a species. And the one is really where we want to look at the phylogeny, at the ecology and things. And I fully support what Svetlana said. You know, this is how we should spend our energy, should be going into really unpacking those, especially if they're important in in terms of functions, etc. But on the other hand, we have not covered the diversity, and we need to create a system where we can actually, when people develop information, produce information, pin that information. And I strongly believe that the name is the pin that keeps information together, because the moment you use the same name when you do a search or whatever, then you start creating that information. So in my mind, I see two ways that will probably go forward. I don't know if it will happen, but the one is where we really put a lot of energy, understand the ecology, work with the ecologists and understand these. And on the other hand, we will have names and they will accumulate information over time. So a description would be not something that is produced by the original person who's made the pin, but it will be a concerted community effort in creating these descriptions. And for that, I think a sequence will probably be the pin that we need, because it's the easiest way of linking things. And as you said, you know, what we want is stability also in terms of names. And therefore, and we've never been able to do it, but we really need a short format, you know, like a little note where you describe whatever characteristics you have. That pin is there. If nobody ever looks at it, it's fine. But you know about the existence of that organism. But once you have a name and there's genome information or a culture, people start seeing that, oh, I find the same thing. It's important. And then people will go and that information will then accumulate. So it will become like a snowball. And I think in my mind, those are the two directions. And I believe we should have a place for both of them.
Right.
Yeah, sounds reasonable, yeah.
Yeah. Having a pin on the map is extremely important so that one knows what to work with and how better and how deep to characterize it. So Preben has just sent in a word that I also see a value of having a genome-based systematic framework, but I understood from the presentation that there is a demand for more and better phenotypic characterization. The physical strain is useful for this, and thus my question. So
Yeah,
just to clarify.
I agree. Yeah. I agree. Mm-hmm.
Yeah. So the presentation was basically, to adding value to a microorganism description. It's not a demand. It's kind of, I take it as a recommendation.
Sure.
Yeah. And I would personally like to do that because That's one of the key things when, as a taxonomist, you are limited to yourself and nobody else wants to read your work other than another taxonomist. And
Yeah, yeah.
that was the starting point where Svetlana started her presentation. That as a taxonomist, if there is only another taxonomist reading your presentation, and that also gets boring at the end, it doesn't make sense to describe only to that limit. So don't limit yourself, explore the boundaries, reach outside the boundaries, and think out of the box. I would really like to, make a point about that experiment you did with the spiral organisms, that you have a tube and you have a petri plate at the bottom. I really enjoyed that experiment that you showed, and whether that was an established method or is it an innovation in the lab that was done.
This is a cheap innovation.
But
Cheap
but but
innovation.
it really tells you something about what the organism is actually doing and
Yeah.
how it mobilizes itself towards methane. So
Yeah.
that's something really interesting.
Yeah, not only this, because we were surprised when we added some beads in there into this semi-solid medium. It
Right.
went even faster because these bacteria need some particles to be around to move in the way they get used to. Can you imagine this? Yeah. So
Right. Yeah. So thank you so much. And there are no more questions, I guess. Thank you, Svetlana, for this excellent session. And thank you, everybody, for participating and posing those questions that, um, taught us how to think beyond just species descriptions. Yeah.
thanks.
Thank you. Thank you.